Hello everyone. In this video we are going to discuss the regulation of glycolysis. In the previous video we discussed the reaction of glycolysis, what was in glycolysis. It involved a preparatory phase and a payoff phase. First of all, glucose was converted into glucose 6-phosphate. Glucose 6-phosphate was converted to fructose 6-phosphate. Fructose 6-phosphate was converted to fructose 1,6-bis phosphate, which was cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and these two were interconvertible, and glyceraldehyde 3-phosphate was converted into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate was converted into 3-phosphoglycerate. 3-phosphoglycerate was converted into 2-phosphoglycerate, which was ultimately converted into phosphoenol pyruvate and then pyruvate. So all the reactions were reversible except three reactions, which are highlighted by red color here. The conversion of glucose to glucose 6-phosphate, it was first irreversible reaction. Fructose 6-phosphate to fructose 1,6-bisphosphate, it was the second irreversible reaction. And the conversion of phosphoenol pyruvate to pyruvate, it was the third irreversible reaction. And these three reactions are the regulated reaction of glycolysis. You should know that the reaction which involves any kind of regulation is a regulated step of metabolic pathway, while what is a commented step, the commented step is the one reaction after which the new product is committed to that pathway. It cannot move back. For and committed step of glycolysis is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, and it is also the rate limiting step. So all three reaction are regulated step. This is also regulated, this is also regulated, and the third one is also regulated. But the rate limiting step is second one, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. It is rate limiting as it is the slowest reaction, and it is also committed step as the fructose 1,6-bisphosphate is then committed to glycolytic pathway. It cannot move into other pathway. And now we move toward the regulation. So we know that glucose is converted to glucose 6-phosphate, and it can involve two enzymes, glucokinase and hexokinase. Hexokinase is basically inhibited by its product. This reaction involves the product inhibition. Product inhibition means that the product of this reaction, glucose 6-phosphate, inhibits hexokinase. When the concentration of glucose 6-phosphate increases, it exerts negative effect or inhibitory effect on hexokinase and inhibits it. While the regulation of glucokinase involves a separate mechanism, glucokinase is not inhibited by glucose 6-phosphate. Instead, it is inhibited by fructose 6-phosphate. Glucokinase is inhibited by fructose 6-phosphate. What happens when the concentration of fructose 6-phosphate in a cell increases? Suppose this is a cell, and this is nucleus, and this is cytosol, and when in cytosol fructose 6-phosphate concentration increases, it causes the movement of glucokinase from cytosol into the nucleus of cell. And in the nucleus of cell there is a protein which binds this glucokinase, and it is glucokinase binding protein. And this glucokinase binding protein or glucokinase regulatory protein binds to glucokinase and keeps it inside the nucleus as long as the concentration of fructose 6-phosphate is high in the cytosol. When concentration of fructose 6-phosphate falls down and concentration of glucose is increased, then this is freed from glucokinase, is freed from nucleus, and it is released back into the cytosol. So what happened here? Hexokinase was inhibited by its product glucose 6-phosphate, while glucokinase was inhibited by fructose 6-phosphate, the next product of next reaction. And how it is regulated? Increasing concentration of fructose 6-phosphate causes the movement of glucokinase from cytosol into the nucleus, where it binds with glucokinase regulatory protein. This protein holds the glucokinase into the nucleus. As long as concentration of fructose 6-phosphate is high, glucokinase stays into the nucleus, and when its concentration decreases, it moves back into the cytosol and regulates the reaction. So it was basically regulation of the glucokinase and hexokinase, and glucokinase is primarily active into liver cells, as well as its function is liver cell and beta cells of pancreas. Now we move toward the kinetics of these two enzymes. Hexokinase is specific for different hexoses, glucose like fructose. Similarly, glucokinase also is specific for different hexoses. It also catalyzes the conversion of glucose, fructose, and other hexoses. But the kinetics are different. Hexokinase has low Km and low Vmax. It has low Km and low Vmax, while the Km and Vmax of glucokinase are comparatively high. It has high Km and high Vmax. What does it mean? It means that Km is the concentration of substrate required to half saturate the enzyme. So the concentration of substrate required to half saturate the hexokinase is small. It is half saturated at small concentration of substrate. It means that it has high affinity for substrate. It binds the low concentration of substrate and catalyzes the reaction. While glucokinase has high Km, it means that its affinity is low. Its affinity is low. It means that it cannot bind the glucose or other hexose at low concentration. It binds the sugar at higher concentration. At higher concentration, it is half saturated. So it means that glucokinase is primarily functional at higher concentration of glucose. It functions at higher concentration of glucose, and hexokinase function as low concentration of glucose mainly. So, glucokinase is basically active in liver cell and in the islets of Langerhans or in beta cells of pancreas. So, what happens? What does it do? It removes the higher concentration of glucose. When the concentration of glucose increase in blood, it is removed by liver cell and the beta cell of pancreas, and there is secretion of insulin. So, it is called as the glucokinase act as a insulin sensor and determines the threshold of the insulin secretion in the beta cells. So what we have studied yet, we have studied the glucokinase regulation and hexokinase regulation. Hexokinase is inhibited by its product glucose 6-phosphate. Glucokinase is inhibited by fructose 6-phosphate. And how it inhibits? Fructose 6-phosphate causes the movement of glucokinase into the nucleus of cell, where it binds with glucokinase regulatory protein. And as long as the concentration of fructose 6-phosphate is high, it stays in nucleus. When its concentration decreases, the glucokinase moves back from nucleus into the cytosol. And the hexokinase has low Km and low Vmax, and it can bind effectively with low concentration of glucose. While glucokinase has high Km and high Vmax, it means that its affinity for substrate is low, and it binds with substrate at higher concentration, and it is acts as the insulin sensor, or plays role in insulin secretion inside the beta cell of pancreas. And how insulin secreted? Basically, when glucose enters in the beta cell of pancreas via GLUT2, it is converted into glucose 6-phosphate, and this glucose by glucokinase, and its glucose 6-phosphate is converted into ATP. Then ATP sensitive potassium channels are closed, and when ATP sensitive potassium channels are closed, beta cell is depolarized, and when it is depolarized, calcium enters into the cell, and calcium causes the exocytosis of insulin vesicle, and insulin secreted from this cell. It is the basically mechanism which is not required here. Now we move toward the regulation of second step, which is the regulation of phosphofructokinase 1. And this enzyme is inhibited by ATP and citrate. ATP shows high energy signal. A glycolysis is basically working to provide the cell with energy. And when there is increased level of ATP, it means that cell has surplus amount of energy. It does not need the further supply of ATP. So increasing level of ATP basically inhibit the phosphofructokinase 1. But keep in mind that ATP is also a substrate for phosphofructokinase 1. So initially, when concentration of ATP increase, it acts as a substrate, and increasing concentration of substrate increase the activity of phosphofructokinase 1. But up to a limit, then the ATP act as an inhibitor of phosphofructokinase 1 and inhibits its activity. Secondly, citrate is involved in TCA cycle, and citrate is also an inhibitor of phosphofructokinase 1. And what are the stimulator of phosphofructokinase 1? AMP actually stimulates the phosphofructokinase 1, as AMP shows the low energy signal in the cell, and it stimulates this enzyme. Further, this is stimulated by fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is a compound which stimulates phosphofructokinase 1, and it is the most potent stimulator of phosphofructokinase 1. And how it works? Basically, Fructose 2,6-bisphosphate is formed from fructose 6-phosphate by separate enzyme phosphofructokinase 2. Phosphofructokinase 2 is a special enzyme which converts fructose 6-phosphate into fructose 2,6-bisphosphate. And this enzyme phosphofructokinase 2 basically has two domains. It has a kinase domain and it has a phosphatase domain. So kinase domain basically performs the phosphorylation of fructose 6-phosphate and converts it into fructose 2,6-bisphosphate. And this kinase domain is active. It is active when it is in dephosphorylated form. It is active when it is in dephosphorylated form. So when it is dephosphorylated, it performs the phosphorylation of fructose to fructose 2,6-bisphosphate, and fructose 2,6-bisphosphate stimulates phosphofructokinase 1 and the glycolytic pathway moves on. When it is phosphorylated, what happens? It becomes inactive, and when it becomes inactive, the fructose 2,6-bisphosphate is not formed. Fructose 2,6-bisphosphate is not formed. As a result, the stimulatory effect of it on phosphofructokinase 1 is removed, and when this effect is removed, rate of glycolysis falls. Now the question is when it is phosphorylated and when it is dephosphorylated. So we should keep in mind that glucagon promotes the phosphorylation of kinase domain, and the insulin promotes dephosphorylation. Insulin promotes dephosphorylation, and insulin promotes dephosphorylation by activating phosphatase domain. Phosphatase domain removes phosphate and activates it. So basically, when the level of glucagons are high, the kinase domain is phosphorylated and it is inactive. When it is inactive, the stimulatory effect of 2,6-bisphosphate is removed from phosphofructokinase 1, and the rate of glycolysis decreases and the consumption of glucose decreases. While when the level of insulin are high, what happens? When insulin levels are high, then it is dephosphorylated, and when it is dephosphorylated, the concentration of fructose 2,6-bisphosphate increases. When its concentration increases, its stimulatory effect on phosphofructokinase 1 is increased, and the overall glycolytic pathway is increased. So this step is also regulated by hormones. How it is regulated? Glucagon promotes phosphorylation of kinase domain of phosphofructokinase 2, and when kinase domain is phosphorylated, it becomes inactive. Fructose 2,6-bisphosphate is not formed. When it is not formed, rate of glycolytic pathway decreases. When insulin is in high concentration, it is dephosphorylated. When it is dephosphorylated, fructose 2,6-bisphosphate is produced, and when it is produced, it promotes the stimulatory effect on phosphofructokinase 1, and all this process becomes speed up. So it is basically the regulation of phosphofructokinase 1. Now we move toward the next step, or third regulated step of glycolysis, and this step is involves the conversion of phosphoenol pyruvate to pyruvate, and it is catalyzed by pyruvate kinase enzyme, and ATP is also produced in this step. So it is basically stimulated by fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is the product of this reaction, phosphofructokinase 1 reaction. So the product of phosphofructokinase 1 stimulates pyruvate kinase. So in this way, the activity of two kinases, phosphofructokinase 1 and pyruvate kinase, is coordinated, and this is regulated as feed forward regulation. It is called feed forward regulation, in which the product of this reaction is basically stimulating the next reaction. And it is how it is inhibited. Pyruvate kinase is again inhibited by phosphorylation, and again phosphorylation is promoted by glucagon. When glucagon levels are increased, pyruvate kinase in phosphorylated form, and conversion of phosphoenol pyruvate to pyruvate is decreased. And when it is decreased, phosphoenol pyruvate goes to gluconeogenesis and produces glucose instead of producing pyruvate. And it means when level of glucagon are high, phosphoenol pyruvate is converted back to this pathway, and then glucose is produced. But when the level of insulin are high, it is pyruvate kinase is in dephosphorylated form, and when it is in dephosphorylated form, the pyruvate kinase is active and it converts the phosphoenol pyruvate to pyruvate, and there is net consumption of glucose. So it is basically regulated in this way. And one more thing is here is glucagon. Glucagon basically conserves glucose, and it conserves and saves glucose, while insulin promotes consumption of glucose. So insulin upregulates, insulin increased secretion of insulin upregulate all three enzymes. When level of insulin remain high, the synthesis of glucokinase or hexokinase increases, the synthesis of phosphofructokinase increases, the synthesis of pyruvate kinase increases, and these glycolytic pathway speeds up. While when glucagon level remain high and insulin level remain low, the synthesis of these enzymes decreases and they are down-regulated. So this is the regulation of glycolysis by different allosteric regulators and hormones. So this pyruvate which is formed here, pyruvate is next metabolized by TCA cycle. If it is not metabolized by TCA cycle, this pyruvate gets converted into lactate. This pyruvate, if it is not metabolized by TCA cycle in anaerobic condition, or if the respiratory chain is not sufficient, not capable of metabolizing this, it get converted into lactate, and this reaction is catalyzed by lactate dehydrogenase, and it involves the consumption of NADH. So the reaction of basically this reaction is also reversible reaction, and its reaction is determined by the level of NADH, or you can say NADH or NAD ratio. If this ratio is high, it means that the concentration of NADH is high, and when NADH is in high concentration, it promotes the forward reaction, conversion of pyruvate to lactate. But if the concentration of NAD is high, then reverse reaction is favored and lactate is converted into pyruvate. In skeletal muscles, mostly pyruvate is converted to lactate and under anaerobic conditions, and this lactate accumulates in skeletal muscle, which leads to muscle cramps. While in heart muscle, in liver, the lactate is used. Lactate is either used to produce glucose, which happens principally in liver, while in cardiac muscle it is used to produce energy. So this is the fate of pyruvate. It is either converted to acetyl-CoA, which is metabolized by TCA cycle, or it is converted to lactate, and lactate is either used to produce glucose by gluconeogenesis in liver, or it is used to produce energy. So one thing we should consider more here, that is arsenic poisoning. One thing more here is arsenic poisoning, which happens and decreases the production of ATP by this pathway. Where does this arsenic poisoning happens? Basically, when arsenic is present, arsenic basically combines with glyceraldehyde 3-phosphate and forms complex with it, and this complex is converted eventually into 3-phosphoglycerate. This step of 1,3-bis-phosphoglycerate is skipped. In the presence of arsenic, glyceraldehyde 3-phosphate is converted into 3-phosphoglycerate, and this step is not involved. When this step is not involved, no production of ATP. As a result, glycolysis happens, glucose is consumed, but there is no production of ATP. So this is the main defect or main poisoning of arsenic which happens in glycolytic pathway. One more thing we should know, this is pyruvate kinase, and pyruvate kinase also, the reaction of pyruvate kinase also produces ATP here. And if pyruvate kinase is not present or deficient congenitally, it leads to deficient or decreased production of ATP in glycolysis, as these two ATPs will not be formed and there will be no production of ATP in glycolysis, and it affects the red blood cells, as red blood cells depend upon glycolysis for their energy requirement, and this energy is required to maintain the normal shape of the red blood cells. And when the pyruvate kinase is defective, ATP is not produced, the shape and cell membrane of red blood cell become defective. As a result, they are phagocytosed and there is anemia. So it was all about today's lecture, in which we covered the regulation of glycolysis, arsenic poisoning, as well as the deficiency of pyruvate kinase. So I hope you will like this lecture. Thank you.